happen infinite screen.
Hi, it's live.
All right, it's live. The defense will start in about 10 minutes. Remind me to exit from that before the closed session. That's not supposed to be broadcast to the world.
What do you mean by closed session? Did you hear it said?
Yeah, like just Paul and the 4 committee members will say.
All right. How's Lexi doing?
Well, we finally got, so yesterday we had that appointment, the Oakland follow-up, and it was the Oakland Marathon, which goes all around the lake and the bed in my house.
Great.
So
It was a marathon to get to.
step one was we figured out how to get to the vet. While we were there, like outside, we were like, "You're doing great!" Like loud rock music. Statements. But we finally got the, temporary ceasefire of like, okay, you don't have to come back until it goes worse.
Okay, that's good.
Which might not be very long, but which is good. And Jesse is sorry to make it. I didn't figure.
It's in the middle of a work day.
Some people do.
Yeah.
Since the first day came, it's taking off.
No one wants to travel so
Hi.
Are they able to like find things? I think like no one else is gonna get married. We get married on like a Wednesday. I'm so excited.
Stream's going good. How's it going? Here we are.
Yeah, to me, someone is doing something that is not like that.
Thanks for coming out. I really appreciate it. Oh, this is just enough to like have married anymore.
Oh, yeah.
Now I'm awake.
Yeah.
Like in bed.
Dating's like freaking out looking at
What does that mean? Let me hear that.
Good. Busy at work, but I'm excited for
Well,
Well, what are we doing? Yeah. We're excited.
oh, just throw some things before we start.
Sure. I
I'm
don't
just
know.
gonna run to the bathroom first.
Yeah. There's a child in your bed.
What did you do? So sleep. Woke up before
Hey.
Make sure the rain.
Oh,
Like
so big.
me and my puppy.
Nice. I do too.
Wow.
She's so chill. Except last night when she was screaming around. We've never seen that. So, she only does it at night. She does it
That's sweet, yeah. Same.
Yeah.
Hey.
Super adorable.
Well, it's no problem.
The restrooms are closed down here. I left them last week when I went to. Yeah, and the second floor too. Did it go? The first floor open.
I'm no longer in meetings. Last hour.
I didn't plan on leaving until we got out. Once we were locked out, I was maybe some. Are you gonna follow? Are you adding 45 minutes? You know, if you want me to.
No, I
You'll
think I
probably
can
need to unplug something.
probably unplug this one and mess things up. How did that laser pointer work okay?
Yeah, it seems to be going good well. So I'll just plug it into the computer when I'm ready to go. And then I think we're still missing 3 committee members.
So yeah, it's not even 10 yet. So I think these morning talks are not so easy to do. It's a Monday morning, everyone's out of it.
And I know that there's a psychs thing at
Yes, which skipped.
Oh, yeah.
It's crazy because like, my mom, I don't know. A little bit better for you. Here. Tell me if you like this one well, number 2. I like it better because it makes you sure. And I love the gray background. It's just like,
that's that's my guy's introduction. So perfect. Yeah, this looks
oh,
great.
this isn't you.
Oh no, that is me. But this is,
Oh.
I didn't prepare this. He did. So
How nice of him. And he asked you for a photo and you willingly gave it.
no, I think I found it.
Do me a favor and don't spill this. Okay, another favor is because you are making the presentation. When you are finished, turn this button off and that will turn off the projector lamp. Sure. It's extremely important because these are really expensive bulbs and we're trying to not use them if we can.
Absolutely.
Okay, so I'm Shelley. I work next door and I kind of manage this space.
Thank you. Thank you.
What's it on? Metabolic?
Mitochondrial and metabolic regulation and differentiation.
I'm sorry. Had a really good time. Where did you go to school?
So I'm just finishing.
No, elementary.
In Wisconsin.
Why do they make us do these things? And so what year was this?
I was in 5th grade.
So what year, like 80s or?
No, this was probably 98.
So oddly, I was born in 1968, so I was in school in the 70s, and this is like the same outfit.
I wasn't particularly stylish, so
Well, who was? Nobody was. No one. Good luck.
thank you.
Take care.
Thank you.
Yeah, it's right. So, yeah, it's sorry. So, that's what it is. Basically, we would take the things that are left tags, purify their interactive proteins, and we do back in. 10. So a high, and I'm sure you're looking at that first. Probably 5 minutes ago, somebody else where
Welcome, everybody. It's a pleasure to welcome you all to the thesis defense for Aparna Bhaduri. In addition to colleagues and thesis committee members, we are fortunate to have Aparna's parents here. We welcome them from sunny Wisconsin, as well as some of the important people that Aparna collaborated with over the years, most notably Alex Singlewickel, who's in the back, who's now a director at Genentech. So Aparna has changed a lot since she entered graduate school on her first day. She's become a lot more relaxed, and she's really developed as an amazing, amazing scientist. So going back, though, here she is planning her first experiment with just common reagents on the kitchen table. And these tended, even at an early age, back in Wisconsin, to be very successful. She produced some pretty amazing results. That cake is, I think, testimony, the multicolored cake is testimony to that. But Aparna matured very early as a scientist and really got into some really exciting things and actually rose to the top of where she was in Wisconsin. And here you can see her wearing gloves. It looks like she's on the roof, engaged in some type of activity there, but she really reached the apex of her community, which is actually the same community that I grew up in. And I know as a graduate student advisor, you're not supposed to, have any favorites, but Aparna and I have shared over these past years something I've never shared with another graduate student. And before the parents get nervous, that's the love of the Green Bay Packers. And as Kerr is also a Wisconsin man, this is something that we commiserated with a lot about and enjoyed a lot. And I always got the injury report from Aparna every morning after the game. It was really a pleasure. Now, in the lab, Aparna has been incredibly productive. This is an action shot captured by Joanna Kowalski of her in-action lab. As you can notice, the biohazard bin is open because stuff is flowing into it so quickly. And if you look at Aparna's bench, it's a model of order and precision. But all that really masks an unbelievable productivity. So, you know, many graduate students have successful careers at Stanford, but you're going to hear a little bit about this body of work today. She's going to share this with you. She has published additional first or co-first author papers, some of which are very bioinformatic in nature, some of which are genetic in nature that she published with Alex as a co-first author. She's also published additional papers. I think she's the only student in the lab to co-author 3 papers in Nature Genetics during her time. So this has been pretty unbelievable. But Aparna has a bit of a partier side to her as well, and she's made great friendships in the lab and has been actually an amazing babysitter, I understand. And here she is with Sebastian Barajas, the son of one of the other students in the lab. And also, I want to give positive idea on this child, and I thought this was Zara. There she is with Zara, who's the daughter of Ashley in the lab. And so Aparna has done some great things. So she's also had an amazing and sometimes long-distance relationship with Care, who's I know been very important to her. And here is Care, who is in the field of education, appropriately in an educational garb. And she's had great relationships with not just people in the lab, with her family or friends. And she's going to tell you about her tremendous work today. So it's a real pleasure to introduce you to Aparna Bhaduri.
Thank you, Paul, for that very kind introduction, and thank you to everyone for coming to my defense today. I'm very excited to share with you my work looking at systems analysis of cellular differentiation and identifying novel roles for mitochondrial and metabolic regulation. So, throughout my PhD, I have sought to use systems-based analyses to understand human biology and disease. And the framework that we like to use to study this process is one where we look at the progression of a stem cell. So here you see how a stem cell is able to give rise to another stem cell, but also to a progenitor cell. And this progenitor cell similarly gives rise to a normal progenitor cell and also to a differentiated cell. This is an important chain of events that is essential to human biology, but it is also interesting because at each stage of this progression, there are things that can go wrong that could yield some sort of disease, frequently cancer, as shown here by a cancer stem cell. Now, while we are interested in all of these processes, my PhD and the work I will share with you today has focused on this particular transition of the progenitor cell to the differentiated cell, specifically in the context of somatic tissue differentiation. Somatic tissue differentiation is extremely important because it's the process by which we give rise to various tissues in our body throughout our lifetime. For example, we can give rise to muscle. If we work out, we want to be able to build more muscle. Bone, if we break a bone, we want to be able to regenerate that bone. Fat, we all have the experience of gaining weight throughout our lifetime. And skin is very important because it protects us from the outside world and is regenerated approximately once every 28 days. I will use the skin as a model for many of the biological studies that we're doing in this talk today. And the epidermis, as we see here, is a key organ that protects us from the outside world, protecting us from insults like sunlight and other environmental factors, and preventing water loss from the body into the atmosphere. The epidermis is comprised of progenitor cells, making it a good model to study this transition. And these progenitor cells give rise to differentiated stratified layers of the epidermis, but also regenerate this basal layer. The epidermis is an excellent model, not only because of these characteristics, but also because it is easy to acquire, meaning that we can do many of our experiments in human primary keratinocytes. Additionally, it is an excellent model for disease. Here we see several examples of diseased epidermis. In the first example, we see an example of epidermolysis bullosa, where collagen 7 or other proteins that anchor the skin to the basement membrane are deranged. In panels B through D, we see examples of ichthyosis, which are frequently a result of mutations in differentiation genes themselves and result in itching and scaling. In E, we see an example of a nevus, which is frequently occurs as the result of excessive MAP kinase signaling. And in F, we see a full-blown cancer, which has facets of this signaling as well as dedifferentiation, and in this case is a cutaneous squamous cell carcinoma. So, the ways in which we are using the epidermis is in two parts in this talk. In the first, we'll look at transcriptional networks identifying mitochondrial regulators of epidermal differentiation, and in the second, we'll look to metabolomics, elucidating a role for glucose in regulating differentiation. So to begin, epidermal differentiation, as I showed you, has these various layers of the epidermis and can be marked by a variety of primarily structural proteins that can tell us where in the process we are, which are useful for experiments. Additionally, our lab, as well as many others, have identified a number of known regulators of this process. As you can see here, there are two examples of non-coding RNAs that regulate this process, as well as a couple signaling cascades. However, the primarily proteins that regulate this process are either transcription factors or chromatin remodelers, meaning they localize to the nucleus of the cell. This is very important to understand gene regulation. However, I was intrigued by the fact that at this very last stage of differentiation, cells enucleate, making the cornified envelope, leading me to think that perhaps there are additional proteins that are not nuclear that are regulating this process. So, I wanted to know if we can identify novel, non-nuclear regulators of epidermal differentiation. And to do this, I wanted to use network biology. Network biology is an idea that you can take outputs at a systems level from a cell and understand relationships between them that you wouldn't otherwise be able to do without the network. In this example, we have nodes represented as circles, which are genes, and edges represented as lines that show interactions between these genes. From this network, we can see that NANOG is very central to the process being studied here, whereas ELYS is less central to this network. Networks in biology can be made from a variety of inputs, ranging from DNA, RNA that will show us the transcripts that are turned on in that cell, proteins, as well as metabolites. For the first part of this talk, although we've looked at many of these inputs, we will focus on RNA transcripts. Looking to RNA transcriptional networks, there are a number of existing algorithms that existed when I began this work. And in a nice review in 2012, a group from Harvard looked at these various network algorithms. These algorithms were primarily transcription factor-centric based network algorithms. And as you can see here, there were a number of methods that were used. What this group did was they tried to combine the outputs from these methods into a community method that looked to improve upon the function of those other methods. On the y-axis here, you can see that the area under the precision recall graph is the authenticity by which they were able to recreate networks that they were looking at. And as you can see here, they're performing decently well in silico networks that are generated by a computer, but drop off when they go to bacterial networks, such as E. coli. What struck me about this was that these transcription factor-centric networks were unable to well recreate the transcriptional networks of eukaryotes in yeast, such as Saccharomyces cerevisiae, suggesting to me that perhaps we need to include inputs that are not transcription factor-centric and may also enable us to find non-nuclear regulators of various processes. So I devised my own method. I wanted to emulate biological networks, so I looked to the scale-free small world network, which is a network that has a shape, as you can see here, that has hubs that are central to the network, but in general, the network is very highly connected. Additionally, a bunch of experimental methods have seen that expression profiles, metabolic networks, as well as protein-protein interaction networks have this topology, suggesting that these highly connected networks are a key facet of biological function. So I devised proximity analysis, where we first begin by rank ordering all of the genes within any data set being considered. This allows us to compare different data set types, such as microarray data and RNA sequencing data. We then take the correlation between any two genes in these data sets, getting a value that we call the proximity value. To generate the scale-free small world network, we use the concept of preferential attachment, which is the way that scale-free small world networks are thought to have evolved. Basically, any gene that is joining the network is more likely to join to a randomized set of genes that are already highly connected, and is less likely to join to genes that are not highly connected. The only modification to this probability of making the preferential attachment network was weighting it by this proximity value in order to offer it biological significance. This gave us a probability for every edge within the network, and then we could iterate thousands of times in order to get a consensus probability for what should go into the network. We could then rank these by probability and take the top n number of edges and make our network. Using this approach, we applied it to the Saccharomyces cerevisiae data and compared it to the outputs that we saw from the existing network algorithms, and observed that proximity analysis was better able to recapitulate experimentally validated edges that are known to exist within the yeast, showing us that including non-transcription factor-centric edges was important in recapitulating this transcriptional network. Given that this approach seemed to work in yeast, we decided to apply it to epidermal differentiation. We began making our correlation matrix with keratinocyte differentiation time course data, 14 data sets across the time course of differentiation, either microarray or RNA sequencing. We then topologically constrained this data and identified the hubs based on genes that were highly connected within the network. We finally applied one last filter of disease relevance in order to hopefully identify proteins that may be essential to various processes, and in this case, weighted the hubs by the degree of change in cancer data sets. Because cancer and differentiation are rather opposite processes, we hope to enrich for potentially disease-relevant genes. Using the dynamic hub score, we ranked all of the known regulators of differentiation and found that the majority of them clustered above the 95th percentile, but also showed us that these were not the only regulators at that level, telling us that we were able to identify non-transcription or chromatin remodeler-based factors as well as the known regulators. When we look to the network pictured here, we can look to the most connected genes within this network and find that they are enriched for gene ontology terms related to epidermis development and epithelial cell differentiation, which gave us a confirmation that our method was at least enriching for things we know to be important. When we specifically rank the genes based on that dynamic hub score, we see that the top 2 genes are already known to regulate differentiation, and a number of others are disrupted in a variety of epithelial diseases. The third gene, MPZL3, has unknown mechanism as well as unknown disease relevance, but was upregulated in differentiation and downregulated in squamous cell carcinoma. MPZL3 was knocked out by 2 separate groups. One of the groups focused on its skin phenotypes, seeing that there were epidermal hyperplasia, hypergranulosis, hyperkeratosis, parakeratosis and wounded edges, lesional skin, club hair, and scales, suggesting that a role for MPZL3 in differentiation was indeed feasible. Another group, interestingly, observed a metabolic phenotype for MPZL3, where the knockout mice were unable to gain weight, had lower blood glucose, and lower fat mass, despite being fed a high-fat diet and having a higher food intake, suggesting that MPZL3 has an interesting organismal role as well. To study MPZL3 in epidermal differentiation, we used an organotypic model of differentiation, where we seed primary human keratinocytes onto devitalized human dermis and raise this to the air-liquid interface, where we are able to regenerate organotypic epidermal tissue over the course of 7 days that recapitulates many of the characteristics of human skin. When we look to MPZL3 in this context, we see that organotypic tissue treated with control siRNA has a very nice basement membrane, shown here in green, that represents the collagen 7, as well as differentiation markers in red that show keratin 1, keratin 10, loricrin, and transglutaminase, with the nuclei shown in blue. But that tissue treated with siRNAs against MPZL3 decreased the expression of these differentiation markers. When we performed RNA sequencing on this data, we see that the downregulated genes were significantly enriched for genes related to epidermis development, telling us that MPZL3 is indeed required for differentiation. We wanted to know how MPZL3 fit with what we already know about epidermal differentiation. So to do this, we looked to data sets of known regulators of epidermal differentiation and saw that its expression was downregulated when KLF4, P63, or COR1, and ZNF750 were knocked down. Moreover, when we intersect the gene signatures of MPZL3 with these factors, we see a significant overlap, but no significant overlap when we intersect with the control regulator of progenitor state. We additionally observed that these 4 transcription factor chromatin remodelers are binding directly at the MPZL3 genomic locus, suggesting a direct regulation. And we identified that MPZL3 is downstream of these regulators by overexpressing MPZL3 in the context of the knockdown of these factors, observing either partial or complete rescue of the differentiation defects. Given that we now understood that MPZL3 was downstream of known pathways of differentiation, we asked, how does MPZL3 regulate epidermal differentiation? And to do so, we wanted to look to proximal interaction partners at the protein level. So we used BioID with VIR-A. In this system, we fuse a promiscuous biotin ligase to our protein of interest and overexpress it in keratinocytes. In the context of excess biotin, reactive bioAMPs are generated and tag vicinal protein interactors. Upon purifying these tagged proteins, we can then identify which proteins they are by mass spectrometry. When we do this, we see that there are 55 proximal protein interactors of MPZL3 that primarily localize to the mitochondria of cells, which led us to ask whether MPZL3 itself was in the mitochondria. Using electron microscopy, we see that MPZL3 is indeed observed within mitochondria of cells, and quantitation of these images suggests that the majority of MPZL3 is in the mitochondria, though there is some in other parts of the cell. Mitochondria are known to regulate a number of cellular processes. Primarily, mitochondria are famous for regulating apoptosis, by which signals from outside of the cell activate caspase cascades and result in death for the cell. But additionally, mitochondria are known to regulate a transition from a pluripotent to a differentiated cell, primarily by structural changes of the mitochondria, as well as changes in their glycolysis and lactate production. So this seemed reasonable that MPZL3 may be regulating this process from the mitochondria. Looking at the list of interaction partners, the top interactor was FDXR, or ferredoxin reductase, which had the most number of spectral counts and the highest probability SAINT score, which tells us whether or not this is likely to be a real interaction. Looking to FDXR, we wanted to validate this proximal interaction using proximity ligation analysis, an immunofluorescence-based assay where we would take an antibody to either MPZL3 or FDXR, and then if the antibodies were close together with rolling circle amplification, we would generate a fluorescent signal. Indeed, we see this signal when there is no knockdown of either protein, but when we knock down either protein, we abrogate the signal, suggesting this is a real interaction. We additionally validated the interaction by co-immunoprecipitation, immunoprecipitating MPZL3 and FDXR and blotting for the other protein. Looking to FDXR in epidermal differentiation, we again use the organotypic model. And when we knock down FDXR, we do see a reduction in the expression of differentiation markers, phenocopying that of MPZL3. We further validated this with RNA sequencing and observed that while FDXR regulates more genes than MPZL3, the overlap between the regulated gene sets is significant and is enriched for genes related to epidermis development and keratinocyte differentiation. Moreover, when we overexpress FDXR in the context of MPZL3 knockdown, we're able to rescue the transcript levels of differentiation markers, suggesting that FDXR may be downstream of MPZL3 in differentiation. So this brings us to the question, what function do MPZL3 and FDXR serve during epidermal differentiation? FDXR is a p53 target gene and is known to be required for reactive oxygen species-mediated apoptosis. Reactive oxygen species are a family of oxygen radicals that are generated during a variety of chemical processes intracellularly as well as extracellularly, and are known to regulate and be regulated by a variety of processes, ranging from normal signaling to oncogenes to hypoxia, with a number of outputs that affect a number of processes. Additionally, it was recently shown that ROS are required for epidermal differentiation. In this paper, a group at Northwestern knocked down a mitochondrial transcription factor, showing a phenotype where there was a decrease in expression of epidermal differentiation marker proteins. But with the addition of galactose oxidase, an enzyme that generates ROS, they were able to rescue this phenotype. They also showed the inverse, where in organotypic raft culture, when they add EUK134, a sponge for ROS, they're able to abrogate this differentiation. So we looked to ROS in our system and observed that by day 6 of differentiation, we have a significant increase in ROS within the system. Additionally, knockdown of MPZL3 and FDXR results in a decrease in this ROS, and overexpression in subconfluent keratinocytes results in a slight but significant increase of ROS within the cell. We wanted to test if mechanistically this was how these MPZL3 and FDXR controlled differentiation, and again knocked down MPZL3 and FDXR, but added back galactose oxidase, and saw significant rescue of transcript levels of differentiation markers, as well as an increase in ROS levels. We additionally saw the opposite. When we overexpress MPZL3 and FDXR in subconfluent keratinocytes that normally would induce differentiation markers, in the presence of the ROS sponge, we reduce these transcript levels, as well as the ROS levels. So, what are the individual roles of MPZL3 and FDXR in regulating this ROS? We hypothesized that it was related to FDXR's enzymatic activity, which catalyzes a reaction using NADPH to reduce ferredoxin, one of the key first components in the electron transport chain. Interestingly, NADPH, the substrate for FDXR, is known to quench reactive oxygen species, and we wondered if simply by removing the substrate, it allowed for the accumulation of reactive oxygen species. However, there was a catch. FDXR levels do not change during differentiation at either the transcript or protein levels. But we do know that MPZL3 levels change, and we wondered if somehow MPZL3 is stimulating FDXR activity. The way that we tested this was by making enzymatic mutants of FDXR, and we observed that while the full-length FDXR was able to rescue the differentiation defects of MPZL3, the enzymatic mutants were not, suggesting that there was a role for FDXR enzymatic activity, and we hypothesized this is related to MPZL3's expression in differentiated keratinocytes. This brings us to our model, where we used proximity analysis to identify MPZL3. We then observed that the transcript of MPZL3 is driven by known regulators of differentiation, and that the protein moves to the mitochondria, where it perhaps stimulates FDXR, and together they control reactive oxygen species to drive terminal epidermal differentiation. While there are still parts of this mechanism that are not well understood, we believe that we now have a new regulator of how a mitochondrial protein could relate to epidermal differentiation. So in summary, we were able to better reconstruct eukaryotic networks with proximity analysis and identify MPZL3. Moreover, we saw that MPZL3 is required for epidermal differentiation, and its interaction with FDXR regulates ROS-mediated differentiation. All right, so to switch gears a little bit, partly stimulated by our interest in MPZL3's phenotype at the metabolic level organismally, as well as the idea that metabolites are another cellular input that we could use to understand the state of a cell, we wanted to look at the metabolome of differentiated keratinocytes. So to do this, we set up a metabolomics experiment, where we used subconfluent keratinocytes to model the progenitor layer of differentiation, day 3 keratinocytes to model early differentiation, and day 6 to model late differentiation. At each of these stages, with 5 biological replicates, we isolated metabolites. We actually sent cell pellets to a company, which then isolated the metabolites and did 5 types of mass spectrometry to identify metabolites within the cell. This resulted in 614 metabolites at any point of differentiation. And in this heat map, you see the relative fold change at either day 3 or day 6 compared to the subconfluent keratinocytes in the progenitor state. When we looked at the metabolic pathways that were enriched in these various days, we saw a couple positive controls. First, gamma-glutamyl amino acids are known to be a substrate for transglutaminase, a key differentiation gene that cleaves these amino acids and offers protective barrier function for the cell. Additionally, we know that lipids are key to also offering barrier function, and we're excited to see fatty acids as one of the primary metabolic pathways that were enriched. We also observed a number of pathways related to amino acid metabolism that have not been well described in differentiation, but might be interesting in future work. The top upregulated metabolite during this process of differentiation was transurocanate. Transurocanate is known to accumulate in the top layers of the skin and has photoprotective qualities, so we thought this also was a positive control. But we're surprised to see that the second most induced metabolite was glucose, which typically is not thought of as accumulating within cells, so we were very intrigued. Glucose, as you might know, is a sugar and is one of the monosaccharides that makes up sucrose, or table sugar. Glucose, whose chemical structure is shown here, can be metabolized in a variety of ways in most cells, and primarily goes through one of three pathways. As you can see, the reactions in the back here are quite complicated, but primarily, glucose either goes through glycolysis, largely to make ATP, pentose phosphate to generate nucleotides, or the hexosamine pathway to make post-translational modifications. All of these pathways must require D-glucose being enzymatically catalyzed by hexokinase into D-glucose 6-phosphate, the molecule that we see here. Once that has been accomplished, glycolysis takes this D-glucose, makes fructose 6-phosphate, and after a series of reactions, generates pyruvate. This pyruvate can go through the less common lactic acid fermentation, which less efficiently makes ATP, or more efficiently be converted to acetyl-CoA, go through the citric acid cycle, which generates a lot of NADH, and then oxidative phosphorylation to make a lot of ATP. Also, glucose can go through the pentose phosphate pathway, which uses the committed enzyme, glucose 6-phosphate dehydrogenase, or G6PD, through a series of reactions to make nucleotide precursors. Or alternatively, it can go through the hexosamine pathway, where the fructose 6-phosphate is converted by GFPT1 through a variety of reactions into a number of post-translational modifications. These are the three primary pathways in most cells, although there are cells, such as liver cells and fat cells, that are also able to generate glycogen from the glucose. So, back to our observation that glucose is highly induced in metabolomics. We wanted to ask, how can we validate that this accumulation is real and not an artifact of the mass spec? So we first began by trying to observe it. We used this analog called 2-NBDG, which, as you can see structurally, is interesting about this molecule is that if you phosphorylate it with hexokinase, it loses its fluorescent signal, meaning that anything that we see is this molecule, not modified and 2-NBDG. And when we look to the cells, subconfluent, day 3 or day 6, treated with 2-NBDG, and this is live cell microscopy, we do see an increase in the accumulated 2-NBDG. We can quantitate this across many images and many cells and normalize the signal to the number of cells per image and see a significant increase from subconfluent to day 3 and day 3 to day 6. However, this is not glucose itself, so we wanted to use other methods to also validate this finding and chose to use radioactively labeled glucose. In this case, we used C14 at the C6 position, which, as you can see from the map down here, will specifically go through glycolysis. When we incubate cells that are differentiating over the entire time course of differentiation in either the subconfluent day 3 or day 6 state, we see a very significant increase in intracellular glucose accumulation. We additionally saw a similar finding when we saw D-glucose that was labeled at the C1 position that specifically goes through the pentose phosphate pathway. We additionally see accumulation when we use FDG, or fludeoxyglucose, that cannot be metabolized through any of those pathways. This suggested to us that glucose is in fact accumulating within differentiating keratinocytes, but we wanted to validate this in organotypic skin also. So we used a fluorescent assay to assay for the glucose, and again, see an increase in the amount of intracellular glucose. This suggested to us that in fact, glucose is accumulating in differentiation, but we wanted to know, is it required for differentiation? To do this, we created organotypic tissue in the context of normal glucose or low glucose. And as you can see here, the low glucose condition not only does not express the differentiation markers K1, K10, or loricrin, but also the tissue does not stratify as well as the normal organotypic culture. Moreover, when we stain for Ki67, a marker of proliferation, we see that there's actually higher proliferative rates in the low glucose condition than in the normal glucose condition. We validated this with RNA sequencing using 2 replicates for each condition and saw that the genes that are downregulated by gene ontology terms are significantly enriched for those related to epidermis development and keratinocyte differentiation. We also validated the Ki67 staining by seeing that the upregulated terms were strongly enriched for things related to the cell cycle. Also interestingly, we saw that many of the known transcription factors that regulate epidermal differentiation were not induced at all in the low glucose condition. And when we look at the transcription factor signatures of these genes with the glucose signature, there is a large overlap of the number of genes that are regulated. All of these are significant. So we tried an experiment. KLF4 and ZNF750 have been well described by Lisa Boxer, a previous graduate student in the lab, to induce the expression of differentiation markers when overexpressed in proliferating keratinocytes. And indeed, that's what we see here. When we have KLF4 and ZNF750 in normal glucose conditions, we see an induction of differentiation markers. But when we overexpress them in the low glucose condition, we do not see an induction of these differentiation markers to the same level, suggesting that not only is glucose required for epidermal differentiation, but also for the function of these transcription factors. Glucose has been described as being required in a couple of other differentiation systems. In myoblast differentiation, it has been shown that glucose restriction results in AMP kinase activation. This activation inhibits glucose uptake and also inhibits myoblast differentiation. In a very nice paper building off of this work last year in Cell, in osteoblasts, it was shown that glucose uptake through the GLUT1 receptor is required for preventing the phosphorylation of AMP kinase and for the accumulation of a transcription factor that is tissue specific, that then drives both glucose uptake and differentiation, suggesting that this is somewhat consistent with what we are seeing in keratinocytes, and also led us to ask the question, is glucose taken up faster in differentiated keratinocytes? Is that how it's accumulating within the cell? So to test this, we used radio-labeled glucose, all 3 forms, and did a 5-minute experiment where we exposed the cell to the tracer for 5 minutes and then harvested the cells and measured the intracellular glucose. Very surprisingly, the subconfluent undifferentiated keratinocytes in all 3 cases were uptaking the glucose about twice as fast as the differentiated keratinocytes, suggesting to us that the idea that it's an uptake-driven phenotype may not be the complete story. We then wanted to look at how this accumulation happens over time, and in this case, used FDG, which cannot be metabolized and therefore prevents confounding variables of changes in metabolism. Again, as we see, at 2 minutes and 5 minutes, the subconfluent cells are uptaking the glucose faster than the differentiated cells, but by 30 minutes, it's almost leveled off, and by 3 hours, the differentiated cells have taken over, suggesting that even though this glucose isn't entering the cell faster, it is accumulating, leading us to hypothesize that maybe in the subconfluent state, it's leaving the cell faster. So to test this, we did a pulse chase experiment. Again, we incubated the cells for 5 minutes with FDG, and we see the same thing where the subconfluent cells are uptaking the glucose faster. But then we also kept some cells where we washed out the FDG, put them into normal media, and saw how much glucose is retained over time. When we do this, we see that the subconfluent cells indeed are losing the glucose faster, whereas the differentiated cells are keeping it for a full hour before starting to lose it, suggesting that this might be an instance where instead of glucose, which is normally transported both in and out, is instead going unidirectionally into the cell. And we'd like to do further experiments to validate that this is in fact what is happening. So the cell has all of this extra glucose. What is it doing with all of this stored glucose? The first hypothesis would be that it is metabolizing it. However, glycolysis rates in differentiation are decreased compared to in subconfluent cells. And when we look at oxygen consumption, we see that there is an increased oxygen consumption during differentiation. This was very interesting to us because it was recently published that in differentiating retina cells, from frog, they were able to see an increase in oxygen consumption in differentiated retina cells and a decrease in lactate, which they used as a measurement for the amount of glycolysis that was occurring. This is particularly interesting because it suggests that the glucose within the cell is not going through the citric acid cycle, but instead there's an alternate source going through the citric acid cycle, resulting in the oxidative phosphorylation that we see in the differentiated state. The glucose is instead doing something else. We looked at this data in our metabolomics and observed that lactate levels, in fact, are decreasing during the differentiation time course. And when we look to all of the glycolysis intermediates, we see that none of them are accumulated at the same rate, at the same amount as the glucose. We then looked to the other pathways of glucose metabolism and saw the same was true for pentose phosphate as well as hexosamine. However, there is a caveat when we're looking at pathway intermediates when looking to glucose metabolism, because frequently when we think of metabolites, we don't think of them just hanging around. They're frequently fluxed. And the hexosamine pathway gives us a unique opportunity to test for the outputs of this entire process, which are post-translational modifications. So we blotted for O-glycosylation, a key post-translational modification that is a result of the hexosamine pathway, and see no real difference in the amount of this post-translational modification, either in normal or low glucose over the course of differentiation. And the signal is normalized to a loading control. So this suggested to us that the cell is not actually significantly metabolizing all of this glucose, and led us to hypothesize, do keratinocytes actually need accumulated free glucose for their differentiation? So to test this hypothesis, we thought that perhaps if we overexpress hexokinase, this committed step in glucose metabolism, we would reduce this pool of free glucose and maybe observe a phenotype. So we did this experiment and indeed see when we look at a variety of differentiation markers in the context of hexokinase overexpression, we don't have the same level of expression, showing a differentiation defect, as well as a decrease in the glucose levels in these cells. We then wanted to make sure that this is in fact a result of hexokinase's enzymatic activity. So we used a competitive inhibitor, 2-deoxyglucose, to inhibit hexokinase. And when we do this experiment, again, we see the differentiation defect at the transcript level. But with 2DG, we see a rescue of both the glucose levels as well as the transcript levels, suggesting that this is indeed an enzymatically dependent situation. However, I just described to you how there's this shift away from glycolysis, so we wanted to make sure that that wasn't a confounding variable in the metabolism of this glucose. And we also overexpressed G6PD, which is one step beyond the hexokinase reaction. But the idea was if we deplete this substrate, perhaps hexokinase will become more active, deplete the glucose, and give us the same reduction in the free glucose population. And indeed, we again see a differentiation defect and a reduction in glucose levels when we overexpress G6PD. We rescued this phenotype with an inhibitor of G6PD. Again, we see the differentiation defect and a rescue of transcript levels and glucose levels with DHEA. We finally wanted to just complete the story and do this with the hexosamine pathway as well. So we additionally overexpressed GFPT1. When we do this, we see less of a differentiation defect. It is 2 steps removed, but we also see less of a decrease in glucose levels. However, the defect that we do see is again rescuable with the addition of a specific inhibitor to GFPT, DON, bringing back the glucose levels as well. So together, all of this showed us that this accumulated glucose does appear to be required in differentiation. And we wanted to know if this is characteristic of other forms of somatic tissue differentiation. So we tested it in 3 other models of somatic tissue differentiation: adipocytes or fat cells, myoblasts or muscle cells, and osteoblasts or bone cells. Using the 2-NBDG, we again do see some accumulation of this molecule between the progenitor and differentiated cells. We then repeated the radio-labeled glucose assays, where we looked at uptake over 5 minutes and observed cells in all 3 cases are uptaking the glucose faster than their differentiated counterparts. But over a longer period of differentiation, we see significant accumulation, particularly in adipocytes and osteoblasts. We don't see as much accumulation in the myoblasts, but we again do see the shift, suggesting that while kinetics may be somewhat different between the cell types, this does appear to occur, this accumulation, in other models of somatic tissue differentiation, leading us to a model of progenitor cells, where you're bringing in the glucose and exporting it quite liberally. When you have the glucose in your cell, you're happy to metabolize it. But in differentiated cells, you don't bring in the glucose quite as fast, but you also don't seem to let it out. And while you do metabolize it, it seems like glycolysis happens less. And this accumulated pool of glucose, therefore, may serve as a signaling molecule. We hypothesized that there might be a glucose-binding protein that is recognizing all of this free glucose and signaling to tissue-specific transcription factors that are driving differentiation. There are a number of questions that we want to answer about this process. Specifically, how does glucose accumulate? How does this transport become unidirectional? And how does the metabolism of glucose slow down? Additionally, we have questions about the kinetics of glucose requirements. Is it a switch that you just need in the beginning, or do you need it sustained over the time course of differentiation? And most importantly, how does accumulated glucose drive differentiation? Is there a glucose-binding protein that is mediating this function? We did set up an experiment to identify glucose-binding proteins, and there are two approaches that we are trying to take. One is a bibliomic approach. We know that there are a number of glucose-binding proteins, either transporters or enzymes, as well as proteins with similar structure. We identified these proteins by taking all proteins that are known to be related to glucose and making a hidden Markov model of their structures, then searching the rest of the proteome to find similarly structured proteins. And from this, we generated a list of 526 proteins. The second idea was to take an experimental approach, perhaps using an affinity binding column, which I will describe now. The idea is that if we can run keratinocyte lysate over a glucose analog, such as dextran, which you can see here is essentially branched chains of D-glucose. And because it's a polymer, it will come in a variety of sizes within the column. When we do this, we can run the lysate over the column and wash with high salt levels in order to hopefully remove non-specific binders. We can then elute with 1 molar glucose and hopefully remove any glucose-binding proteins that are on the column. As controls, we can use other monosaccharides, such as fructose, so that hopefully we can identify glucose-binding proteins, not just sugar-binding proteins. And we can use a different type of column, such as saccharose, which is a polymer of agarose, which has quite a different structure. With all of these controls and the results from what we get off of the column, we can use mass spectrometry to identify the proteins. And in fact, we did this experiment in triplicate and identified 326 proteins that within each replicate passed the filters with its controls. As you can see, there's some but not substantial overlap between the replicates. However, we were okay casting a wide net in the hopes that maybe something would emerge in 1 or 2 of these replicates. Additionally, of importance, we did identify that 3 key proteins, which are known glucose binders, were in all of the replicates: hexokinase 1, hexokinase 2, and SLC25A1, which interestingly is an intracellular citrate transporter, but is known to be sensitive to glucose levels and regulate insulin signaling. When we look to the gene ontology terms of these genes in the heat map, they're primarily enriched for translation elongation and splicing. We don't know if this is important to the process or if it's just stickiness on the column, but we're hoping future experiments will help us disambiguate which of these proteins may be true glucose-binding proteins. When we do, with this list of 326 from the experimental approach and 500 some from the bibliomic approach, we can perform a CRISPR screen, where we design 6 guide RNAs for each of the proteins being queried. We can then package them into lentivirus and infect keratinocytes that are highly expressing Cas9, and then select for integration, allowing us to specifically cut 1 gene out of 1 cell at a time. We can then grow out these cells, either in subconfluence to look for genes that are essential for the progenitor state, and replication and survival, or we can differentiate the cells. When we differentiate the cells, we can then replate them back into subconfluence, in order to identify colonies that specifically would emerge from cells that didn't differentiate in the first place, and hopefully help us identify genes that are necessary for differentiation and the cell cycle arrest that is key for this process. We can then amplify these guide RNA cassettes and sequence to identify the target genes. In summary, we observed that glucose accumulates in differentiated keratinocytes and is required for epidermal differentiation. We further observed that it appears accumulated glucose itself is necessary for differentiation, and that the glucose is accumulating in other examples of somatic tissue differentiation. So with that, I have many, many acknowledgements to make, and I can't possibly name everyone individually. But I first and foremost must thank Paul, who's been an excellent mentor, especially for these slightly off-the-wall projects, as well as Alex Ungevikkel, who is an incredible mentor during the entirety of the MPZL3 project and who consulted with basically every experiment in that project, as well as Ariana Sanchez, a graduate student who was in the lab, and Angela Guerrero, a medical student, who have done an amazing amount of work on the glucose project and helping me move that forward. I also would be very remiss if I didn't acknowledge Gayatri Gauri-Shankar from the Gambier lab, who has helped me immensely with all of the radioactivity experiments, as well as my committee, Howard Chang, Mary Tarul, Tim Stearns, and thanks to Julian for being my chair, as well as the epithelial biology program labs, the Oro lab, Chang lab, Marinkovich lab, Wang lab, and Lee lab, which make this an absolutely amazing department to work in. Everyone is super helpful, and the administration that keeps everything going, the Pamela Bernstein, Luana Morcom, as well as the Cancer Biology Administration, Grace and Andrea Inamato, who was chair of the program for most of the time, as well as my funding. I'll be now happy to take questions, after which I'll have some brief personal acknowledgements. Yes.
So you characterized 3 major pathways downstream of glucose, and yet you didn't mention glycogen. I'm just wondering if one of the things that you could have considered in the pathway towards keratinocyte differentiation would be the accumulation of glycogen, because that could increase against chromatin, for example.
True. So the reason we didn't look at glycogen is that the enzymes that are canonically thought to make glycogen are not expressed in keratinocytes, and there was no glycogen in our metabolomics. But we do think that that's an important pathway for many other cell types, and potentially that would be a good enzyme to overexpress in order to deplete that pool of accumulated glucose. Yeah, so I do know that they have identified it with other systems, but it's true that especially in the keratinocyte where there's lots of lipids. Yeah, absolutely, we can do that. It's a good idea. Yes? So this is an interesting question that we haven't looked at specifically. We do know that insulin pathway signaling is important to keratinocyte differentiation. However, the other systems where glucose uptake has been looked at, where it's required for myoblast and osteoblast differentiation, have actually had their uptake for that differentiation be insulin independent. So I don't know in keratinocytes if those two are related, but that would be an interesting line of experimentation that we could go down.
For cool down of the NPZ,
Mhm.
you use that technique. What's the advantage of this one? Regular beats that you couple with the antibody.
With the Bure?
Yeah, magnetic beats. Regularly, you can use the magnetic beats that
Mhm.
you put the antibody and cool down the targets.
Mhm.
What was the advantage of this?
I'm sorry, I'm not exactly getting the question. Of the Bure technique?
Yeah.
Yeah, so the reason that we did that was because conventional immunoprecipitation was very challenging for MPZL3 because it's a transmembrane protein. And this was creating problems. We were having to do pretty harsh lysis, and then we weren't getting very much material on the other end. So the Bure technique was a really nice technique that allowed us to see vicinal interactors. Yes?
Great talk. Question about MPZL3 and proximity analysis.
Uh-huh.
So can you sort of walk us through why that scored highly in the proximity analysis? What were the scale of networks that actually affects the most?
Mm-hmm.
That's scored positively.
So I think that's an excellent question. And what's really nice about that approach is that it allows us to find things that aren't necessarily increasing hundreds of fold, But in different data sets where there's more subtle changes, we see that it's traveling together with a lot of different proteins and a lot of different proteins that seem to be central to the network. And so it's basically things that are changing moderately that wouldn't be at the very top of a simple fold change list would be associated with the protein and then, or with the transcript and then moving forward.
So the mitochondrial, ribosomal, RNA polymerase type proteins would then score higher than other things because they have a lot of friends. Is that the idea?
Well, so what's interesting is most of those proteins are very stable in transcript level, and one of the weaknesses of the proximity analysis is it can't find regulators that aren't changing. So for example, FDXR is not at all highly scored in the proximity network. So a lot of those things, for good and worse, have very stable transcript levels during differentiation, so we don't necessarily see them to be highly enriched, but there are some structural proteins that may or may not be functional, and I think some of them have been studied more recently, that are thought to be non-regulators that now seem to be traveling and scoring more highly. So, yes. Thank you.
So people that have diabetes,
Mhm.
they accumulate glucose,
Mhm.
and then what it has been shown is that you accumulate reactive oxygen species
Mhm.
inside the cell, and you start modifying all the proteins and mess up everything. What is showing the first part is that for differentiation, you're accumulating reactive oxygen species, and then in the second part, you're showing that you're accumulating glucose.
Mhm.
So you think that that is the right direct connection. That when you accumulate glucose, you're just accumulating reactive oxygen species which connect to the first part of your problem.
That's a great connection, and it's very interesting and something that we've recently started thinking about. And it might be, I know that there are skin phenotypes that are associated with diabetes, and we don't know if we have an exact connection here, but potentially this could be relating back to insulin signaling in some way. This could be relating to uptake in some way. I don't know how keratinocytes are changing their behavior in the presence of that basically bath of glucose. And so it's a good question, and it's an interesting connection, and we don't quite yet have a mechanistic hypothesis for how that might be working, but I think it's something that we need to think about more. Yes?
Are you looking at blocking efflux of the differentiated cells? Just because it's, you look at like, things that could be blocked. You're seeing the accumulation.
Right.
Because of changing those kind of.
So we haven't yet found anything, and this is very interesting because we were just considering ways in which that you could be making this efflux unidirectional versus bidirectional, and we know that there are ways that you can post-translationally modify these transporters. The transporters can also cycle within the cell, but we don't have a great method for doing that yet. So first what we want to do is really validate that we're not getting the glucose getting exported, and then we're going to look into various ways of either inhibiting that process or seeing if there are modifications of those transporters. Yes?
Very interesting hypothesis. I was wondering from your pull-down experiments with the dextran columns and so forth, did any of the conserved proteins that you identified in other samples appear in your network?
It's a good question. I don't remember off the top of my head. I haven't actually gone back and looked, but it's a good idea to check that out. A number of those things that came down with the column are not significantly changed in their transcript levels during differentiation, but then again, there's a population of them that are, and so that population would be a good candidate for something that might be highly connected in the proximity analysis. All right, I briefly have a few personal acknowledgements, so if you'll just bear with me for a quick second. So first, I absolutely have to thank my mentor, Paul Kavari. He's been the most amazing PI. And those of you who know Paul well know that there are two very strong characteristics about him. One, he's extremely optimistic and he's extremely persuasive. And I would say that without these characteristics as a mentor, I would not have done nearly as much as I did during my PhD. He convinced me I was capable of doing things I totally wasn't, and then he pushed me and I was able to do them. So I owe him many thanks for all of the success that I've had as a PhD student. And in that vein, I also have to thank Alex Ungevekel, who I believe just had to step out. But he was the most amazing mentor and took all those great ambitions that Paul instilled in me and made me a good scientist who occasionally used controls. And without him, I would be nowhere in my PhD. I also have to thank the entire Kvari lab. Through the years, they have molded me as a person and a scientist, have given me great ideas, given me great perspective on life and in science, and I'm so thankful to them. Those of you who are in the Kvari lab know that it's frequently chaotic, and much of this I would attribute to the large number of graduate students who have come through the lab during my time there. And I have to thank each and every one of them. They've been like family to me. I imagine this is what it's like to have a lot of siblings. I'm an only child, but essentially all of them, there's always someone around to either entertain you, go for coffee with, talk to, talk science with, talk intellectually about politics or something completely different, learn about sci-fi. There's always someone willing to engage in some way at any time of the day, night, or weekend. And so I have to thank them. And there are three of them I have to thank particularly individually. Ashley, who is one of the busiest people I know, but has always had time to help me, whether it's mice or whether it's setting up the stream for this talk. Brooke, who has pushed me as a person, scientist, and runner, and who I miss dearly, as well as Joanna, who really is the glue of the lab and keeps the entire lab running. Without her, I don't think that we would have any reagents in lab, which means none of us would do anything. But also, she's been the most important person and comrade during my PhD, who has been an aggressive realist, but also a very kind cheerleader, and has really kept me grounded and given me tons of entertainment. So thank you to Joanna. I also have to thank a number of friends near and far who have supported me and encouraged me through this process, specifically Rebecca Zarin-Pass, who was my family outside of lab for many years. I ate with her, ran with her, talked to her, spent all of my free time with Rebecca for many years of my PhD. I also have to thank Kayer, without whom I definitely wouldn't be here today. I probably wouldn't have eaten in the last few months, because he's great at not only feeding me, but listening to every little experimental detail and occasionally suggesting experiments that I should do. And he has helped me grow as a person. He's taught me compassion, patience, and I'm so excited to marry him. And finally, last but not least, my parents. I have to thank them for teaching me the meaning of persistence in their daily life and for helping me become the person that I am today. And thank you to all of you for being here.
So there'll be a gathering at about noon in the large conference room on the green side of the second floor of CCSR if you'd like to join us there.